Amino acids made me care about OChem. When I first learned about organic chemistry, I was learning a lot of different chemicals and reagents and other things that were never going to happen in the human body because they were so toxic. So it was very hard for me to engage. I knew it was a prereq for medical school, but I didn't really think it was going to be useful for anything until I got to biochemistry. The way enzymes function is based on how they are able to bond, both with themselves in order to create a protein structure, but also with other molecules in order to function as an enzyme. All enzymes are made up of amino acids. So before we talk about actual protein structure, let's talk about an amino acid structure. When we focus on amino acids, the majority of the bonds occur around one specific carbon known as the alpha carbon. It's called the alpha carbon because it's attached to the carboxylic acid functional group, which is the highest functional group. So it's the first carbon attached to it, it's alpha carbon. This carbon is both something called a chiral center, which has to do with light spinning, and a stereo center, which has to do with the direction in which the functional groups are placed if you draw this out. Drawing out structures based on their chiral center or stereo center will also help you be able to imagine what direction other molecules are going to have to come from in order to make bonds with these amino acids. In the human body, all amino acids are oriented in an L position, which means the amino group is to the left of the carboxylic group. In this molecule, you see the carboxylic acid group is circled in red and the NH3 group is circled in blue. And you can see that NH3 is on the left of the COOH group. So this is an L-oriented amino acid. Because all amino acids are oriented in the L position, it makes it easier for peptide bonds to form within ribosomes. It's really difficult to make proteins without ribosomes, so you can also think of ribosomes as enzymes that catalyze the formation of peptide bonds. When peptide bonds are formed, a transition molecule is created in the P site of a ribosome, where the tRNA molecule is still attached to the amino acid, and that amino acid is now also attached via a peptide bond to the peptide chain of other amino acids. The formation of that peptide bond also results in water being lost, which means this is a dehydration synthesis reaction. You are synthesizing a new molecule, the peptide chain, and you are dehydrating or losing water as a result. And the creation of peptide bonds is the primary structure of a protein. The peptide bond is formed from a carbon bound to a nitrogen. And because those have different electronegativities, another way of saying the electronegativity is that the nitrogen is going to take the electrons more of the time than the carbon does, which means there's more moments for the carbon to want to bind with other things because it's missing its own electron. They're going to create a polar covalent bond where the nitrogen is delta negative because it has a higher electronegativity, and the carbon becomes delta positive because it has a lower electronegativity. This sets up the secondary structure of the protein from hydrogen bonding between the polar bonds. If you ever have a proline within a peptide chain or the primary structure of a protein, the nitrogen from the amino group is also within the ring of its R group. So it's got a lot going on already. It doesn't have any ability to create hydrogen bonding. The secondary structure is going to have to bend or lean around the proline. So proline as an amino acid is in favor of leans in the rest of the peptide chain, or it's proline. So proline is pro-lean. In summary, primary structure of a protein is created when peptide bonds are formed between the amine group of one amino acid and the carboxylic group of another amino acid. The secondary structure of a protein is created from hydrogen bonding from the nitrogen and carbon within the backbone of the peptide chain of the amino acids.